Okay, good morning, all of you. So, in the last class, we had looked at the transcription mechanism. How does it happen? Now, we are going to look at what happens after the transcription process. So, that is what is exactly known as the post-transcriptional processing of the RNA transcript. And whenever it says RNA transcript, it usually refers to mRNA, which is like a process, not process, the unprocessed RNA. So the originally transcribed RNA molecule, they are called as primary transcripts. So that means they are hnRNA, that is heterogeneous nuclear RNA. Heterogeneous because they have introns and exons. That's why heterogeneous. And they are biologically inactive. This is the one of the most important criteria. Why biologically inactive? Because we have looked at the structure of mRNA. It needs to have that methylated guanine, it needs to have poly-A tail and so on. So many different components. So these components or these parts or structural aspect of the mRNA, they are very essential for the activation of mRNA. Otherwise, the mRNA will not be able to function. And that is when the RNA transcript or the primary transcript, which is synthesized after the process of transcription, is not biologically active. And because it contains this exons and introns. So these RNA transcript, they undergo extensive changes to become functional or to become biologically active. And this particular process is known as processing of RNAs, or it is also called as post-transcriptional modification of RNA. Later, when we study the translation process, you will also see post-translational modification of proteins. So just be aware of these two different processes. This one is post-transcriptional modification of the RNAs. And during this process, the large RNA precursor is cut into smaller RNAs by what we call as ribonuclease. And this particular process is cleaving. Cleaving as in, like if it is a long stretch of the mRNA molecule or the primary transcript, and which is not just one structural gene but rather a combination of it, then they will be cut into respective RNA fragment, and that action happens with the help of ribonuclease. Again, this ribonuclei or ribonucleic acid is nothing but your RNA, and ASE at the end, that means it's a breakdown, and this P is one of the form of this ribonuclease, and that process is cleaving, which is nothing but breaking it into smaller pieces. Now, the smaller RNAs does not necessarily always mean one particular gene is being cut into different smaller fragments. We already know that, for example, tRNA is there. And we have also studied during the tRNA description that tRNA molecules, they are polycystonic. That means within one stretch, there are many molecules of this tRNA. So then they have to be cut down into smaller RNA fragments to the respective tRNA segment. So that all those processes, they need this cleaving part. Not necessarily always the regular mRNA transcript also need cleaving. So that is not always the case. But surely the second point, that is unwanted ribonucleotide, or what we call as intron, they are removed by nucleases, and this process is called as splicing, or it is called as RNA splicing. So let me show you, where is that diagram? Yeah, here it is. So for example, you can see this is the DNA molecule. This is the start of the mRNA transcription process. And then it undergoes some modification like methylated guanine and so on. After that, the exons and introns are there. Now you can see all the dark or black colored fragments. These are the introns. They loop it up, and then they are cleaved or they are split from the main fragment, and what they get at the end, is only the coding part of the DNA. That is what is messenger RNA, the final messenger RNA. And this poly A tail is also added later, which stays with the mRNA molecule. So this is like a functional mRNA molecule. So this part, this removal of the intron with the nuclease is called as RNA splicing. There are again various mechanisms. How does it happen? There are so many protein complexes which are involved in RNA splicing, but we won't be looking at that as of now. So that is the process of splicing with the help of nuclease. Now, the functional regions, or what we call as exons, they have to be joined. So when these introns are removed, there are gaps in between the two exons, and these gaps, they are kind of rejoined with the action of ligase. Ligation means addition or joining to each other. So that addition of exon or rejoining of the exons happens with the help of enzyme ligase, and that is the process of union. Now, certain nucleotides are added at the end of the mRNA transcription enzymatically, and that is called as terminal addition. Specifically, in case of tRNA, it has these uridine derivatives or uracil derivatives, if I can say so, right? So those derivatives or those modified nucleotides, they have to be added enzymatically, and that process is called as terminal addition. So these are all different post-transcriptional changes. So this terminal addition is one of them. That is, after the synthesis of tRNA molecule, certain nucleotides, they needs to be replaced or added within the stretch of that RNA molecule, and that is terminal addition. Molecules may fold on itself to assume a proper shape. Again, in case of tRNA, it has to go into that L-shape looping, right? So that also happens, especially in case of tRNA and also in case of rRNA, but rRNA is more in association with other proteins, right? So there they resume the shape of those ribosomal structure. So that all of this folding or three-dimensional structure development, that also happens after post-transcriptional, after the post-transcription. Some nucleotides may be modified. Again, the same in case of tRNA, they can be modified even further, like methylation or hydroxylation, depending on what are the changes, whether activation or inactivation is required. They needs to be modified after they are already present in the nucleotide strand, that is RNA strand, and that happens in the case of tRNA. Okay, are there any questions here so far? I hope these parts are clear. Yes.
So the certain nucleotides that are added at the end, that's the poly A tail, right?
Yes.
Okay.
Means that is one of it. In case, so in some of the cases, when the mRNA has to be deactivated, there are this DNA methylation that happens with the help of certain protein complexes, and because of that, the mRNA transcript also get changed to some extent in terms of addition of nucleotide. But again, these are like two different processes. But yeah, one of the example is surely the poly A tail addition. Okay, so then the processing of the mRNA primary transcript. So now we have received the mRNA transcript, which is a primary nature. So these were different changes that happens. That does not mean all of these changes happens in one mRNA transcript, right? Depending on what type of mRNA or tRNA or rRNA it is, these changes will happen. Like for example, rRNA, that is ribosomal RNA, may not need splicing and so on, right? Or may not even need ligation, or may not need a terminal addition. Folding will be required for it. So depending on which type it is, these different processes will happen. So most of the prokaryotic mRNA, they usually do not need any modification most of the times, or even if they need, that's very little modification again in terms of structural arrangement and so on, but they are relatively very simpler, like we had seen yesterday itself. So prokaryotic mRNA, they require little or no modification. But in case of eukaryotes, these primary mRNA transcripts, which are called as hnRNA, heterogeneous nuclear RNA, these needs to be modified chemically in the nucleus itself before they are transported out to the cytoplasm for the protein synthesis process, that is translation. And what are those changes? Splicing, that is removal of introns and joining of the exons. This is done by nuclease, and joining is done by ligase. Capping at the end of 5' end, which is addition of methyl guanosine triphosphate to the 5' end of hnRNA, and this is required for the mRNA structure. And this is one of the like what we say, when the translation happens, we will see this. Why is this methylated guanine or guanosine is so important with the mRNA? So that is the capping phenomenon. And tailing, which is addition of poly A tail. So 200 to 300 adenylate, that is adenyl or adenine residues, they are added at the 3'. So these are the major changes in the mRNA transcript. Here we are not talking of tRNA or rRNA. This is very specific for mRNA, which we have already seen in general. So these are the three different processes that happens for making a functional mRNA molecule. Now, so again, processing, what is this? Oh, again, so there are different ways in which this can happen, right? For tRNA as well as for rRNA. This one, sorry, processing of mRNA. Now this is processing of tRNA. In tRNA, trimming of the ends by cleavage of the phosphodiester bond. Now trimming of the ends, in the sense, you know, tRNA has a specific requirement in terms of 5' end and 3', right? 3' end has its hydroxy molecule, and 5' end has its phosphate molecule. So that way, the requirement, and then because they are in continuous chains with the other RNA transcript, this trimming of the ends, especially at the phosphodiester bond or phosphoester bond, has to happen. Splicing for removing introns. Like I mentioned earlier, that sometimes it may not be there, sometimes if the introns are there in that particular DNA transcript, this splicing will occur. Obviously, it has to happen if there are introns. Addition of some terminal sequences. Addition of some terminal sequences, you again remember there was the CAA. Sorry, not CAA, CCA. That sequence is present at the terminal end of the tRNA molecule, so that the amino acid can go and bind to that particular acid, which was known as amino acid arm or receptor arm, right? So that one, not receptor, acceptor arm. So that was the addition of terminal sequence. And heterocyclic base modification, usually by methylation. Heterocyclic base modification, it refers to the like methylation of the bases, and that is what leads to the conformation in the changes, and that is when it leads to different what we say, looping and so on. So that is the processing of the tRNA transcript. With rRNA, it is very simpler. It just trimming to the appropriate size, whether it's 28S or 20S or 5S and so on. So depending on that particular sedimentation coefficient, they are trimmed to their respective sizes. So that is the, and again this action will be done by nucleases. So these are the different processing of different RNA transcript. Okay, any question here in any three of these processing part? Okay, so if not, then we will move on to the genetic code. Now we already know what genetic code is, right? This is the triplet codon that we talk of, which is present in the DNA molecule. So there were quite a few scientists which were looking at this structure. So how they came to know that it's a triplet and it's not a duplet or quadruplet or any other random combination or so on. So this George Gamow, he was a physicist. In 1954, he indicated that there are 4 nitrogenous bases, but they code for 20 amino acids. And that's when the mathematical calculation they came into picture. So each codon should have a combination of 3 nitrogenous bases. And why that 3? Because if we take 4 into 4 into 4, this cross is additional here. So that is the combination of 3 bases would generate 64 codon. That is 4 into 4, 16 into 4, 64. So that way it will generate 64 codons from these 4 base pairs, but all of these 64 in together, they will code for 20 amino acids and so on. So that was the first study which was done by this George Gamow. Then Crick and Benner, they demonstrated that codon consists of 3 nitrogenous bases with the experimental setup. So they took some specific what we call as nucleotides, specific nucleotide sequences, and then they put it into the culture medium, and then obviously those cells, they took it up this, and then they identified by sequencing how many nitrogenous bases are there in each of these amino acid chains. Nitrogenous bases in amino acid chain associated with the amino acid. So that matching was done, and that's when they came to a conclusion that there are 3 nitrogenous bases. Then the protein synthesis in a cell-free medium by this Marshall Nirenberg, it helped in cracking the genetic code or deciphering the codons of different amino acids. So the progression was identifying the triplet, proving that it is 3 nitrogenous base in a triplet or a codon, and then the next step, which was shown by this Nirenberg, was the which codon codes for which amino acid. So that work was done by this Nirenberg, and I guess he was also given a Nobel Prize for that work. Then this Severo Oshoa's work, it actually discovered the polymerization of RNA with a defined base sequence in a template-free medium. That is the enzymatic synthesis of RNA. Template-free medium, as in it does not have any template as such in terms of additional external source. Whatever is present within those cells, that is what is acting as a template-free medium. That means there is no template-free. It also means a unique template. There is only one type of template present in that medium, and then the enzymatic synthesis, which is more of a like a chemistry experiment rather than a biology experiment, that was done, and he deciphered the genetic code. Now this is the link that I have kept here with this Nirenberg name that describes whole of this experimental setup, but we need not go again. If you at all you are interested, you can go and read them. So Nirenberg, he synthesized the mRNA molecule consisting only of uracil. So when the mRNA had only uracil, the tRNA, which codes for UUU, or the anticodon which will code for AAA, that will go and bind to that uracil and will bring one amino acid. Say next codon will again be UUU. So same tRNA molecule will build on the same what we call as amino acid. So whatever polypeptide chain you get at the end was of only one amino acid, and that was phenylalanine. And this way, Nirenberg identified that a codon which codes for UUU is code for phenylalanine. Similarly, he did for different possible combinations, and that way he deciphered different or identified different codons for the amino acid. So that is just one small example that is given. Yes.
But how did he do it in the cell-free system? Like if there's no cell, then there'll be no ribosomes, right?
These are enzymatic reactions.
Yes, but you'll still need a ribosome, right? Or use something else.
In a way, yes, but within the bacteria, it's very much possible to do it that way.
Okay, so he did it in a bacterial cell.
I assume so, because in a eukaryotic cell, it will be very complex otherwise. I'm not
So,
sure, but
like this can be used to make medicines, right? So you can just create that particular type of RNA that you want to create. That'll create a specific protein, or you can create a new combination of protein.
no, I didn't get it. How what do you mean by it can be used for medicine then?
So it's like this. So everything is made of proteins, right, in the cell. So let's just say that to fight some virus or bacteria, you need a specific type of protein. Yeah, let's say an antibody. So that protein will have a sequence of amino acids, right? You can make the RNA for that and feed it to the cell, and then every cell can make that antibody,
Right.
and then use it.
So in that method, the problem is of antigenicity, right? That is one problem. Like specifically in case of antibodies, not always we can get the success rate of when we add RNA. For sure, we have seen that RNA molecule, specifically mRNA, has a lifespan of hardly 2 to 4 hours, right? So that will not even go to the next generation of cell. Now the next problem comes is if we integrate it within the genome sequence, right? That might have its side effects, like because we are adding something externally into the germ cells and that we are propagating to all the different cells. But that is very tedious process as well. Like especially now with the CRISPR-Cas, it is possible, but earlier days it was not at all possible by them. So the technical challenge is how to make it sustainable. Like you said, it can function. If it is mRNA, surely there is going to be a degradation of that mRNA by the time we inject within a particular cell itself, right? If it is a DNA, it may or may not always get integrated within the human genome, and even if it gets integrated, it may or may not get transferred to the different generations of the cell. Those cells, they might be considered as foreign cells and they will be killed off. So these are the general problems of even these RNA vaccines which are being used. It needs quite a lot of processing of that RNA, the RNA vaccines, and then we have to go for it. So it's not like so simple process as well. Okay, so I guess we will move forward. So this was the genetic code, and again these are the two diagrams. I have nothing else to say here apart from, so there is a sequence UCAG, that is the second letter, and this is the third letter on this side. So you take first of each, first, then second, and then so on. So that way, what you get is different amino acids. And then you have to remember which amino acid codes for which codon, or rather which codon codes for which amino acid, the other way. So these are different what we call as codons or genetic codes for the amino acids. There are short forms which are being used for this amino acid. This also you need to remember. Like this is again relatively very easy. For example, arginine is ARG, alanine is ALA. So that's usually the first 3 letters of the like name of the amino acid. It just in some of the cases they are little different. For example, tryptophan or asparagine, there are little different, but most of them you will easily figure out by looking at the name. So these are different amino acids. Now, so there are certain essential features of this genetic code. Is what is the requirement of becoming a genetic code from that nucleotide base sequence. So first and foremost, it has to be triplet, and quite a few of these we already know. So it has to be triplet. Genetic code is comma-less. That means no punctuation or space between the codon. So we do never write like you must have seen like whenever I write on a board, I write ATG, then space, then CGC, then space. So I write sometimes like that, but that is not the ideal way of writing at all. When we write a genetic code, it has to be in sequence without space, without any comma and so on. So that is the general way. But yeah, for understanding purpose, if we are writing something, that is perfectly fine. But genetic code otherwise does not have any space or any punctuation in it. It is non-overlapping. Non-overlapping in the sense the one first triplet code or first 3 nucleotide bases will not overlap with the second triplet or second codon. These will be separate. So then first 3, then 4, 5, 6 will be the next codon, then 7, 8, 9 will be the next codon. So that way it is non-overlapping. And if had it been overlapping, then it will be like, let's say first 2, 3 will code for one amino acid, then 2, 3, 4 will code for another amino. That would be overlapping, but that does not exist in case of any living organism. So that's why the genetic code is non-overlapping. Genetic code is degenerate. That means more than one codon for a particular amino acid, and that is what is called as degenerate system. That means there is only one amino acid, but there can be more than one codon for that amino acid, and that's why it is called as degenerate code or degenerate system. Now, degenerate here does not literally mean that it gets degraded or something like that, but it just means that it has more than one codon for a particular amino acid. Now, major degeneracy that occurs at the 3rd position, which is the 5' end of the triplet codon of the like genetic code, right? And this 3rd base is described as a wobbly base. Like I had mentioned it earlier, that there is something called as wobble phenomena or wobble hypothesis. So the tRNA anticodon can wobble at its 5' end, that is the last base pair in the sequence, and by pairing with the non-complementary bases. Non-complementary means A will not pair with U or C will not pair with G. And there we had also seen those inositol and modified base pairs, they can also take part, and that is why it can also wobble. That means it is not a perfect match of the nucleotide pairing, and that is what is known as the wobble hypothesis. And this was identified again by this guy Crick in 1966. So that is the wobble hypothesis or wobble phenomenon. Why does that happen? How exactly it happens? Like I will share one article which describes about it, but it is not in the syllabus, so we can skip that part for the class, but I will share the reading material around. So that is the wobble hypothesis. Genetic code is unambiguous and very specific. That means once, let's say ATG is one of the code from the triplet codon, it will always code for methionine. irrespective of the organism, irrespective of its position in the gene or whatsoever. ATG will always code for methionine. And similarly, all the other genetic codes, they will code for the same amino acid anytime, anywhere, right? Even across the organism species or different organisms, they will always remain same. Collinearity, same linear sequence of bases and amino acid. That means, like if the DNA starts with ATG and so, then amino acid will also start with the corresponding codons will correspond for the respective amino acid. So the sequence of the amino acid will also be same. So that is what is collinearity. That means whatever codes in the DNA, it will be replicable in the protein or polypeptide, and it will also go in the linear fashion. It does not happen that whatever is 3' end in the DNA will come to the at the start in the protein molecule. That does not happen. No exchange of the codon sequences happen. That is what is collinearity. That means it remains the sequence of DNA as well as the amino acid remains same. And genetic code is universal. That means whole of the organisms, they have these 4 bases, they have the same amino acids and so on. So there is no difference in any of these things across the different organisms. So that is these are the essential features of genetic code. Any question here? Anyone?
So, like, I had a question, but it might not be very related to this.
Ask.
Like in a cell, like if that's like in a zygote, that's a single cell stage of 2 different organisms. Let's just say a human and maybe an insect. So, like the difference in the cell, is it only in the nucleus or is it also in the cytoplasm?
Cytoplasm also has little bit of differences in terms of proteins that they have. See, like I said, though we have certain proteins which are evolutionarily conserved, and the meaning of these last 2 words is that means they have remained almost same starting from the primitive species to the most complex species which exist on Earth now. So that is what is evolutionarily conserved. So in some of these species, this conservation can vary. And we had seen, I don't remember when and where we had seen this, but we had also seen, I guess in terms of histones, the percentage of match between evolutionary conservation. Some proteins, they are matching with the human histone proteins or any other proteins to 97%. In some protein cases, it might be just 30 or 40%, right? So that way the protein conformation will be different. Function might remain to some extent similar, right? Because ultimately function is decided by the domains or the segments of the protein which are present in it, right? So if at all these proteins are different, then obviously the cytoplasm space or other organelles will also be different. Again, organelles are different in the sense that they might have less number of ribosome, different types of ribosome, or mitochondrial structures will be small, Golgi bodies will be less. So that way the differences will always remain. And like I said, also in terms of proteins, they will surely be different. So that is the case.
So if you take a single cell zygote of some other animal, let's say a chicken, and just the DNA, you take out the DNA and put human DNA in that, so that won't grow up to be a human.
It might grow. It might grow, Pratham, because I'm not sure how much percentage of that experiment will be a success rate, but surely it can grow. Yeah, let's say you try that 100 times, I'm sure one of the times this experiment will be a success. That is for sure. But again, like that number depends on how efficiently we do it, how do we transfer this whole of the genetic material from human cell to chicken cell. So that also matters a lot. But yes, theoretically or even practically it is very much possible. It just the success rate will be very low.
Okay.
Because remember, whole of the information is present in the genes. It is not in the cell surrounding. Cell surrounding, that is like a like we know that endosperm that works with the seeds, it just acts as a nutrition, just as an additional factor. Whatever has to be done by the embryo cells. So similarly in this case, whatever has to be done by the DNA molecules, right? And that will give rise to the mRNA and proteins and so on. So that way I guess it will be what we say, quite possible in terms of it's happening. Okay, so I guess we can move ahead with uh, oh yeah, sorry, one point is remaining in the essential features of genetic code. Again, this part we know for sure. There is something called as initiation codon, there is something called as termination codon. AUG is the initiation codon, UAA, UGA and UAG, these are the three termination codons, or what we call as stop codons. And they have specific names as well, that is Auger, the opal and the amber. These are the three names which are again given by some scientists. I don't know whom and when, but yeah, these are the three names of these three specific stop codons or termination codon. And these both of these, either initiation or termination codon, they are known as signals. Signals for what? Signals to start and signals to stop the transcription processes. And this phenomena is known as punctuation. Punctuation of the genetic code, it is called as. Because in like how we have a first letter capital of the sentence that says okay, this is the start of the sentence, and full stop acts as okay, this is the end of the sentence. So that is like a punctuation for English language. Similarly, the punctuation for genetic code is the initiation codon and the stop codon. So that is one additional feature that we missed out in terms of genetic code. Now again, in case of gene mutation, we have seen this enough in the last chapter itself. What is substitution mutation? Which leads to the sickle cell anemia. That is one of the example of substitution mutation, where one of the nucleotide base is changed, and because of that, one specific, not any random, but one specific nucleotide base is changed, and if that is changed, it can lead to the sickle cell anemia. Frame shift mutation, again the codon has a certain frame, so 3 3 3 3, and if you remove one by addition or deletion, like you add one or you remove one, that will change the codon frames, and that is what is frame shift mutation. And ultimately what you get in terms of protein sequence after this frame shift mutation might have non-functional proteins altogether. So it has also happened in quite a few cases that you know, within the same DNA fragment, let's say in the first codon and the seventh codon, first it has added something and seventh it has deleted one nucleotide. So ultimately, and if these first to seventh codon, they are not really essential for the protein function, then it will not lead to any changes in the protein at all. But yeah, if at all there is any changes that leads to the functional aspect of the protein, obviously that is a harmful mutation. But there are these different possible combinations of this mutation. Like I said, one getting added and one getting deleted within a short stretch of DNA, that may or may not really affect the protein formation at the end. And in terms of importance of mutation, we have studied this. There it is important for variation across within the species and across the species. It is the part of the evolution. Like for example, this Ancon ship. Again, you can Google this. It is like quite a lot of information available. So due to the mutation, the species was generated and then it is evolved. And now in agriculture, all your genetically modified crops, they do have certain mutations, and that is like kind of like what we say, debatable whether they are good or bad, but yes, that is one of the aspect in terms of mutation. Animal husbandry, again the same, for getting more milk or more eggs and so on, or more meat, certain mutations have been done into these animal husbandry things, right? So that way, mutation is important in industrial microbiology, again, to get some more of the antibiotics or use of these microbes for certain purposes. There have been certain mutation which have been incorporated into these microbes. So these are different. Again, I assume these are very simpler things, so we did not spend a lot of time in discussing about this in the class. Then okay, so that is the all of it, the end of transcription. Now the next process, how we have time, 48, yeah. So we will just have a quick overview of the translation, and then tomorrow we'll start with the mechanism of the translation. So translation in general is referred to as protein synthesis. It has different steps in it. For example, activation of amino acid, then attachment of this activated amino acid with the tRNA so that it forms a certain complex, and then this tRNA will go and take it to the ribosome molecule. It will attach there based on the sequence recognized from the mRNA transcript and the anticodon present on the tRNA, and of course the binding sites and so on. It will keep on adding the amino acids again in the same sequence, like initiation, that is from the first codon, then elongation, it will go and elongate, and then the termination. So one of the interesting fact that we will see later, this termination thing, like you might wonder that there are the 64 total codons, and for stop there are, out of these 3 are stop codon. So what do you think, will there be any tRNAs for the stop codons? Any random guess?
Could you repeat the question? Like, I didn't get what you mean.
I can't hear you, Pratham.
Sir, could you repeat the question? Like, I didn't understand.
So there are 64 codons, and out of 64 there are 3 which are stop codons, and for individual codons or individual amino acid there is a particular tRNA. So I asked, what about the stop codon? Do you think they will have a tRNA?
Well, no.
Yeah, so that's, they don't have. And also, if you remember earlier we had seen that tRNA does not tRNA, the stop codon does not code for any amino acid, so ultimately they will not have any tRNA as well for those codes. And because of that, when the termination, like whenever there is a stop codon and no tRNA can come and bind to that particular sequence with the help of tRNA, so obviously that is the end of the protein synthesis. So this process we will see a little detail in later parts. And then the modification of the released polypeptide chain. So whatever polypeptide or protein is synthesized, that is modified in terms of its primary, secondary, and tertiary structure, and that is what is used for the functional aspect of a cell. Now there are different raw materials in general which are used for protein synthesis. Again, this, most of this we know: ribosomes, of course; amino acids, of course; then mRNA and tRNA. mRNA will act as a coding strand for protein synthesis. tRNA will help in bringing amino acid together. Then there are several enzymes, including amino acid activating enzyme, which is specifically called as amino acyl tRNA synthetase. So this is one enzyme which is responsible for activation of amino acid. Like even in case of transcription and replication, we have seen activation of the ribonucleotide and activation of deoxyribonucleotide. That is addition of phosphate group and so on, right? So something similar happens at the first stage. here with the help of enzyme. That is, this enzyme amino acid tRNA synthetase will activate this amino acid, and then there is second enzyme which is peptide polymerase system. This is again a complex of enzyme which adds the amino acid one after another, and it forms the bond between them. So that is, these are the two main enzymes. Then ATP, which is required for the amino acid activation, and GTP, which is another energy molecule. It is used in the peptide bond synthesis. How exactly? Again, that we will see briefly in the later part. So these two molecules, ATP and GTP, they are required. Soluble proteins, these are specifically the initiation and transfer factors. Again, we will see what these factors are. These are certain protein molecules which are required for very specific actions while the translation is happening. And then inorganic cations like magnesium, potassium, and ammonium ions. So that is the requirement of the translation process. I guess we will stop here. In the next class, we will start with this mechanism of the translation. Any doubts so far in this? Any of these? I mean, there is nothing much, but still, if at all you want to ask something. Okay, so if not, then let's stop here.